Hello and namaste everyone. Uh, my name is Ashish Prasad. I'm from Research Institute for Bioscience and Biotechnology here in Nepal. Today I'm here to chair the monthly Biomedicum webinar series. I'd like to express gratitude to our guests and participants for joining us in our Biomedicum webinar series. As a housekeeping rule, I would like you all mute your microphones during the presentation. And the presentation will run for 30 minutes, and after the 30 minutes, 15 minutes will be allocated for the question and answer session. We are very pleased to welcome our speaker for today's webinar, Dr. Raghav R. Poudyal. He is a senior scientist at Pfizer, Cambridge, Massachusetts, specialized in mRNA therapeutics. He accomplished his PhD from University of Missouri, Columbia in biochemistry in 2016. And he was also postdoctoral fellow in the Pennsylvania State University in Pennsylvania. His backbreaking work and dedication on the research has granted him numerous grants and awards. He had worked on numerous research topics which led to the publication of his work in high-impact journals. His research interests are in vitro and in vivo RNA structure function, in vivo evolution, artificial cell, and synthetic biology, RNA, and nucleic acid-based therapeutics. We are pleased to listen to Dr. Raghav R. Poudyal talk today, and I will request Dr. Raghav Poudyal to start his talk.
Thank you, Ashish, for the kind introduction. And also for Cribs for inviting me to this talk. म चाहिँ नि सकेसम्म I wanted to speak in Nepali, तर कतिपय technical termहरू चाहिँ नि इङ्ग्लिसमै भन्दा सजिलो हुन्छ होला, त्यही भएर I think it's going to be English for the most part. So okay, let me get started. त्यो screen sharing चाहिँ नि disable छ जस्तो अहिले. I can't share my screen.
Could you please enable the screen sharing for our guest? And one thing I would like to address everyone, our participant, please, if you can if you can on your video, it will be very helpful for us in the presenter. The engagement will be very impactful for our webinar series. I request our participant, please, if you could on your video.
No, I can't.
Okay. तपाईहरुले देख्नुहुन्छ है मेरो स्क्रिन?
Yes, sir. We are seeing yours.
Okay. Actually. Okay, let's get started. Thank you very much, Aif, again for the very kind introduction and also for inviting me. तपाईहरुसँग मेरो इन्ट्रेस्ट अनि मेरो कामको बारेमा कुरा गर्न पाउँदा चाहिँ एकदमै खुसी लागिरहेछ. So today I'm going to talk a little bit about some of the work I have done over the years, mostly मेरो postdoc area ko, and the topic that I'm going to talk about is how RNA, which is mostly thought of as an informational molecule, right? हाम्रो DNA बाट RNA मा transcribe हुन्छ अनि त्यसपछि RNA चाहिँ translated हुन्छ protein मा. That is like the most commonly thought of function of RNA molecule, but it turns out that RNA can do much more than just be an informational polymer. And I'm going to take you a little bit about some of the themes in origins of life and life kasari start भयो होला about 4 billion years ago पृथ्वीमा, and what is RNA's role in that, and then also tell you a little bit about how we're using mRNA-based therapies, to tackle pandemic-like situations. So, to get things started, yeah, so today's agenda, which I just told you about. So origins of life, right? Like I think one of the most intriguing questions as a scientist is, of course, thinking about how life on Earth started and whether we can find life at other places. And but before we can really understand or start to ask those sort of questions, we need to understand how life works in Earth, right? And we know that modern biology works by your all the proteins that perform your functions in cells are encoded by your DNA, which is transcribed into RNA, and then that gets translated into protein. But if you look at the building blocks of these different molecules, for DNA, you have deoxynucleotides. For RNA, you have ribonucleotide, which is very similar to DNA. So when the DNA gets converted into RNA, it's a transcription because it's sort of similar language, right? You still have nucleotides. But when the RNA gets made into proteins, now the building blocks are completely different, right? It goes from nucleotides to amino acids. So that's why it's called translation, because the alphabets are different. And the way that life works as we know it is that all of the functions in your cells, all the metabolism is mostly driven by protein enzymes that catalyze chemical reactions. So this is something that you may have already seen. This is sort of a pathway map of all the biochemical reactions that is going on in our cells right now that keeps us alive, right? So it's a very highly networked biochemical metabolic network, essentially. And you probably can't see there, but what I pointed out with this arrow is first few steps of the glycolytic pathway. So if you ate some glucose containing breakfast, I guess, in Nepal time, then it gets into this glycolytic pathway, converted into pyruvate, and then fits into the citric acid cycle, which is shown here. And if you really look at life at a molecular level, it is really a bag full of chemical reactions, right? So so to really understand how life formed on Earth, we need to sort of start understanding how we can find chemical catalysts that can catalyze these sort of chemical reactions. So you sort of run into this problem when you think about origins of life and how the first life formed, because अहिले कस्तो हुन्छ भन्दाखेरि चाहिँ अब modern biologyमा चाहिँ you have your DNA that gets transcribed into RNA and that gets translated into protein, right? But then to copy the DNA, you need the protein. Whereas to make the protein, you need the DNA. So which came first, right? Was it the chicken or the egg? So you have this problem, right? So the first cell, what would it have? So it turns out that RNA can actually do both. It can act as an informational polymer and also as a catalyst. And this was actually discovered, early 80s by Tom Cech and Sidney Altman, and they won the Nobel Prize 1989. And what they essentially said was, RNA can not only act as an informational molecule, but it can also catalyze chemical reactions. So if you thought of the very first life, if it had RNA, then it would not need DNA and protein. It could sort of do both the functions with the same molecule. So basically what that enabled was this hypothesis, which is called the RNA world hypothesis, which basically says that, well, we are here now, present life, which uses DNA and protein. So about 4 billion years ago, the cells, sorry about that, let's get there. So yeah, about 4 billion years ago, the cells essentially used RNA as both catalyst and also as an informational polymer. Now, there are also other hypotheses, such as, there can be other polymers such as threose nucleic acid or even deoxynucleic acids that can act as catalysts. But I think the RNA world hypothesis is a nice testable model for the origins of life, because you can really make RNA in the lab and start testing different hypotheses. So yeah, I mean, RNA started out about 4 billion years ago, and it is still very relevant now. And to my Nepalese audience, I guess it's more like Raja Samal, right, who's apparently always relevant throughout the years. So to get things started, how does RNA get its function, right? And it really has to do with the idea that it can fold up into three-dimensional structures. But I mean, RNA is very similar to DNA if you look molecular structure, right? It has the nuclear base and the sugar phosphate. The only difference being, instead of 2' hydroxyl for the RNA, the DNA only has a 2', I guess, a deoxy at that position. And what really RNA does is it can really fold up into three-dimensional shapes. For example, this is the three-dimensional structure of the ribosome, which is the machine that makes protein, decoding mRNAs. And what you can see here is that in cyan and in gray here, it is basically depicting the RNA component of the ribosome, and the machine that really drives life by making proteins, which is the ribosome, is mostly composed of ribosomal RNAs. So RNA is really at the heart of even modern biology as well. And the reason it can actually have that functions is coming back to the structure. So we've found RNAs that can catalyze small reactions, such as cleaving other RNA molecules or binding to other small metabolites. But these guys, George Smith, Jack Szostak, and Jerry Joyce, they invented a technique called in vitro evolution, which is essentially being able to do evolution experiments in test tubes. And what that enabled us was really discovery of RNAs with brand new functions. So to give you an idea of how this technique works is you start out with about 10 to the 15th unique RNA sequences, and you essentially impose them for under certain selection pressure. For example, in this case, if we wanted to identify RNA molecules that bind to ATP, we essentially incubate these RNAs with ATP resin, which is basically an agarose bead that is linked with ATP or any adenosine molecule. So any RNA that is able to bind to the column is essentially reverse transcribed and amplified, and then you can repeat this cycle of selection, right? So in the end, what happens is that you enrich for RNA molecules that bind to specific ligand. And this is just an example. And this particular RNA, it folds up into a secondary structure as depicted and binds to adenosine monophosphate, for as an example in this particular case. And people have really used this technique of in vitro evolution to find RNAs with brand new functions. So to give you an idea of some of the functions that people have discovered, I really want to bring this theme of like how RNA is made, right? Like if in modern biology, we have protein enzymes that replicate both DNA and also RNA. So one of the biggest questions in this field is, are there RNA enzymes that can also synthesize RNA molecules from the very basic building blocks? And turns out, there is. This particular RNA molecule is actually able to extend a primer that is linked to a template, which is shown in this black line here, and is able to actually make RNA molecules without any protein enzymes. So this is RNA making other RNA molecules. So these sort of in vitro experiments and in vitro evolution based RNA enzymes have really allowed us to test different hypotheses that are sort of related to origins of life, whether you can make these biopolymers in the lab, or how these biopolymers perform catalysis, or what does it take to make these interesting RNA enzymes. So overall, where are we in terms of the field, right? Like can so there are like, you can sort of think of this as several layers of questions, right? Can RNAs adopt three-dimensional structures and catalyze chemical reactions? We have numerous examples of this. So, I mean, yes, it can certainly do that. And can RNA be made without any protein enzymes? I think we are, getting there, right? There are some examples where RNA can make other RNAs without protein enzymes. We can make RNAs from building blocks, as I'll share some of the results from my work and also from the others. And that is also being addressed. But really what is really interesting to me is, can metabolic reactions be compartmentalized like cells, right? Like, can we form cells with RNA and these interesting chemistries, just like how biology uses cells, right? I think that is something that is very interesting and has somewhat not been explored as some of the other themes. And of course, as I shared in an earlier slide, there's like this huge network of biochemical reactions in our body, and that's how life works. And what ultimately we want to see is whether RNA can also catalyze chemical reactions in sort of this pathway and really have that network of chemical reactions. So if you look at the interface of this like top-down and bottom-up approach to studying origins of life, right? Like, you can start with a living system and then sort of like break it down into smaller parts, so you end up with a minimal cell presumably, and then also the non-living components, which are essentially metabolites and protein enzymes, all the way in the bottom. Whereas you can also go bottom to up, where you can start assembling these small metabolites and protein enzymes and try to make a minimal cell, and then ultimately lead to a living system. So that's like two ways of studying this sort of synthetic biology or origins of life. But regardless of which pathway you take, you would ideally want to form polymers of functional RNA molecules from monomers. So monomers of nucleotide form into polymers, and ideally you want some of them to have functions. You want them to be catalysts. And then at the end of the day, you want to make sure that all of these different components of metabolites are somehow compartmentalized, right? Because that is very important to life, because we have cells everywhere, all the way from microbes to humans. So so there has actually been some work done in terms of trying to compartmentalize RNA molecules and interesting chemistries that RNA molecules can do. And a lot of the work in this has been led by Jack Szostak lab at Harvard, and now at University of Chicago. And essentially, this is a polymerization reaction where an RNA molecule is being polymerized with a new NTP, or nucleotide triphosphate. And this is a very basic chemistry, SN2-like mechanism, where this 2' or the 3' hydroxyl attacks the phosphorus, and this bond, the oxygen on this phosphorus leaves, and you essentially form this new bond between the extended RNA molecule and this new NTP. So that's how the polymerization works in terms of modern biology and most of the protein enzymes. And turns out, with a little bit of trick, you can actually do this chemistry without any protein enzymes at all. So for example, in this particular case, there's a primer, which is shown here in black. And then it basically base pairs with this template, which is, again, an RNA molecule. And it has a lot of C's in this template. And if you have these system and give a nucleotide, which is somewhat modified in this alpha phosphate. So instead of a nucleotide triphosphate, you have this phosphorimidazolide nucleotide. You actually don't need any protein enzymes. The RNA essentially is able to add those G's right across from these C's, right? And essentially you make this double-stranded molecule where you have made RNA molecules without the aid of any protein enzymes. And Jack's lab has actually done quite a bit of work trying to compartmentalize this whole reaction inside a artificial, essentially vesicles, right? So we're really trying to get to these membrane-bound, I guess, if you want to call it protocells, they're not quite cells yet, but they're sort of, trying to get to a cellular state, but they're like protocells with the membrane browns, that are fully membrane bound, and then contain these RNA primers and templates and these nucleotides, and are able to actually make polymers of RNA. So we can form these artificial cells and actually have these RNA molecules being made. But my focus was something interesting, in a sense that these particular membrane-bound compartments, they're highly sensitive to metal ions, and charged metals or charged molecules are very difficult to transport within these vesicles, right? But in our cells, of course, there's a lot of the times there are protein transporters that help with transporting molecules in and out of these vesicles. So what I was really interested in during my postdoc year was this new idea of membraneless compartments. So these would essentially still compartmentalize molecules, but they would lack a proper membrane, right? And how does that work? So this is a very simplified diagram of what we call complex coacervates, which are membraneless compartments. And really at a basic level, if you have polyanion, which is essentially a polymer with a negatively charged, that is negatively charged, it has a lot of negative charges, and then polycation, which is a polymer with positive charges. And if you mix them together at a certain pH, certain temperature, you start forming these droplets that contain high concentrations of polyanion and polycation. And they tend to also concentrate magnesium, nucleotides, and RNA molecule. For example, in a simple case, this polymer called polyalylamine is highly charged with, it has got positive charges, and it's very densely positively charged. And ATP is a small molecule, but it is also highly negatively charged. So it has 4 charges. And if you mix these two molecules together, you end up forming these nice droplets that have high concentrations of these different metabolites. And turns out that these coacervates or complex coacervates can actually be made by lots of different polycation and polyanion combinations, which I won't have time to go into the detail. But what we have been sort of thinking is that these complex coacervates can potentially compartmentalize nucleotides and magnesium. So they concentrate these molecules, and that can lead to a non-enzymatic generation of RNA polymers, right? Because that is obviously important from origins of life perspective. If you want to have RNA enzymes, you want to first make longer RNA molecules, and we think that complex coacervates may help with that by concentrating nucleotides and other cofactors. And once you do make RNAs, you have these functional RNAs that can have catalytic activities. So these can now drive the metabolic reactions, which can have complex functions, right? Just like how cells work, right? Cells have protein enzymes that do complex chemistries. We think that these complex coacervates, which are these membraneless compartments, may be able to also compartmentalize RNA molecules and have sort of the same function. So to get things more technical, I guess, we tested several different complex coacervates, which are membraneless compartments, and we used different polymers of positive charges, such as oligolysine, which is made with lysine, 10 repeats of lysine, and that has positively charged as well, and some other polymers for the polycationic part. And then as far as the oligo anion goes, we used oligoRNA because RNA also has negatively charged backbone, right? So when you mix these components together, you form droplets, as I showed earlier. And what we found is that if you incubate it, labeled RNA with these droplets, then you can actually see that the RNAs end up in these droplets, right? The fluorescently labeled RNAs are encapsulated by these droplets. And what is really interesting is that you can get huge enhancements of concentrations. For example, we added only 0.5 micromolar of this RNA molecule, and inside the droplets, you can see the concentration has increased quite a bit, like over 100-fold enhancement of concentration. So this is something that is really exciting, and we wanted to explore this further, because if you can encapsulate these molecules within these compartments, then potentially you can use them as bioreactors as well. I'm not going to go into too much detail about this, but what we were able to show is that we could not only form these droplets, but we could also do this non-enzymatic RNA polymerization using this phosphamidazolide activated guanosine molecule. Again, the idea is the same. You have a primer, you have the template, which essentially, adds G's to this primer. And what you can see here is that in the supernatant phase, which is, outside the droplets, you don't really see a lot of polymerization of this RNA, but within the droplets, especially with this PDAC polymer, you can see quite a bit of polymerization of RNA within these complex coacervates. So this was very interesting that we could not only make these membraneless compartments, but we can drive formation of RNAs inside these membraneless compartments. And all of this work is actually already published in Nature Communications. So if you want to learn more about this, then certainly reach out to me, or if you can find the paper, of course. We've also, used this system to activate RNA enzymes. So this particular RNA molecule shown in black is actually an enzyme. It binds to the substrate, which is in green, and cuts the substrate right here, indicated by this red arrow. So what we thought was that we could perhaps use these membraneless compartments as bioreactors, right? Because these membraneless compartments can enhance concentrations of molecules, we thought that perhaps we could use these to bring the enzyme and the substrate together and activate the reaction. And that's basically what we see here. Without the complex coacervates, you see very little activity of the enzyme that cuts the substrate. But as soon as you perform this reaction in the presence of these droplets, you see the enzyme is able to quite robustly cleave the substrate. So this was the first time that was demonstrated that these membraneless compartments could be used to activate RNA chemistries. And we only tested one of the compositions, but it is really, you can use lots of different compositions of these complex coacervates to drive this chemistry. I'm going to switch gears a little bit here. And, I've been talking about membraneless compartments that we have explored in the lab, which are completely artificial, but there are also membraneless compartments within the cell itself. Like our cell has this outer membrane, which is, I guess is somewhat the cytoplasmic membrane, basically separates the cytoplasm with the exterior. And there is also the nuclear membrane, which separates the nucleus, right? And both of these compartments are membrane bound, but within the cytoplasm, there's actually a lot of membraneless compartments as well. And these membraneless compartments are essentially formed by very interactive, I guess, associative interactions between RNAs and different proteins in the cells. For example, positively charged proteins can interact with negatively charged RNAs. There are of course protein-protein interactions with intrinsically disordered regions. There's a lot of hydrogen bonding and other molecular interactions that sort of make this giant hairball of membraneless compartments. But what is really interesting about this field is that RNA is actually able to make these membraneless compartments by itself without any help of the proteins itself, right? For example, there were several studies published, I guess, late last decade, where different labs showed that you can actually, RNAs can form these droplets or compartments that seemingly don't have any membranes by themselves, right? And all of these experiments were done in vitro, but there have also been experiments that have been done in cells that sort of show the same thing. What is really interesting about these different systems is that, well, first of all, in this particular RNA, there is a lot of repeats of CUG and CAG, right? And if you know from your basic molecular biology, that's the C base pairs with the G, right? You have the GC base pair. So you get this extensive interactions between G and C, which makes these RNA droplets. So what we thought was, what about RNAs that are not, either G quadruplexes, which form highly specific structures, or these RNAs that have extensive repeats of C's and G's, like can they also form these membraneless compartments? So as a model system, we use bacterial riboswitches. So riboswitches are RNA molecules that are able to change their structure and regulate gene expression. These are really elegant regulatory elements in bacteria. And usually what happens is that they bind to a small molecule, and then upon binding to the structural molecule, or upon binding to the small molecule, it changes its structure such that either the ribosome is not able to translate, or the transcriptions are of the gene that it is regulating is turned off or turned on for some of the switches. So they are essentially like an on-off switch for gene expression in bacteria. So there's nothing really special about these riboswitches. We just use them as models. But what we were really curious was trying to mimic the cytoplasmic condition. And yeah, and we were trying to mimic cytoplasmic condition because within the cytoplasm, the environment is quite crowded, right? And we tried to mimic that by using polyethylene glycol, which is a very common crowding agent. And what we see is that, without any sort of additional protein enzymes or other proteins, these RNAs are actually able to form these nice droplets. And what we see is that if you label the RNA with a 647 dye and PEG, which is the crowder, with a different dye, you can see that the droplets are entirely composed of RNA, and really the crowding agent doesn't quite get inside these droplets. So what is really interesting is that RNAs are able to form these droplets on their own, and they tend to exclude these crowding agents. So what we were really curious was that you can form these droplets, and we were curious if these droplets actually protected RNA from nucleases. For example, T1 nuclease is a protein enzyme that is able to cut RNA. So if you incubate an RNA molecule with this T1 nuclease, it is basically going to degrade the RNA apart, right? Like the RNA is going to fall apart. But what we thought was, what happens if the RNA is actually inside these condensates, right? Like you have this protected RNA, presumably because it is within this membraneless compartment. Is the nucleus is still able to degrade the RNA? So without the nuclease, you can see, whether you have the condensates or no condensates, the RNA is fully intact. But once you add the nuclease, you can see that if you don't have the condensates, the RNA rapidly gets chewed up, just like what we know what happens with nucleases. But when you form the condensates and then challenge it with the nuclease, the RNA is actually protected much better than having no condensates at all. So although we have not quite explored this, I think this is going to be something that is potentially useful for mRNA therapeutics, because we need RNAs to be stable and more persistent when we use them for vaccines or other biologics. So a summary of this part of the talk: membraneless compartments can sequester RNA and enable prebiotically relevant chemistries. RNA condensation is driven by both extrinsic factors, which is like environment, and also intrinsic factors. I didn't get to talk much about the RNA sequence and structure aspect of things. And really, we can use RNA structure prediction to engineer these condensates. And what we found really interesting was that RNA condensates can really protect RNAs from nuclease degradation. So in the last part of my talk, I I wanted to touch briefly on something that is very interesting, and sort of this new, which has brought RNA field to this new limelight, which is mRNA-based vaccines, right? Which has been sort of at the center of stage because of the COVID pandemic. And I work at Pfizer, and I've been really lucky to be there because we were one of the first companies, I guess, along with BioNTech, and Moderna, to bring in the RNA-based vaccines for the COVID pandemic. Just very briefly, some of the advantages of the mRNA-based vaccines over traditional vaccines is, of course, traditionally, it takes dozen or even longer from discovery to getting your final vaccines. Of course, that has been sort of a historical, I guess, process. COVID was somewhat like a exceptional case because it was a pandemic situation. But we had the vaccine from discovery to having vaccines. It was I guess around 1.5 years or even less than that. And the reason we were able to actually scale this so fast has to do with a lot of the processes that go into vaccines production, because with mRNA molecules, once you have that one process that works for one RNA, it tends to be sort of the same for all the RNAs, because it's the same cell-free processes. Whereas for traditional vaccines, you obviously have to either their protein components or their entire inactive microbes. So every sort of vaccine candidate is different. So there's different processes for every unique vaccine. So that is sort of this advantage that mRNA has over traditional vaccines. And they're really, I guess, in my opinion, there are three main modalities of mRNA vaccines, which is your conventional mRNA molecules, which has where your biologic of interest is translated by the proteins in your cells. You have the self-amplifying modality, where you have your vaccine antigen that is actually amplified by viral replication machinery that actually makes many copies of your target mRNA molecule, which again makes more protein. So in principle, the self-amplifying RNAs are more potent than your conventional mRNA molecules. And you also have your circular RNAs, which is sort of like this new emerging idea, which tends to have more stable mRNA molecules in the cells. But really, there's a lot of things that I think still needs to be addressed. For example, how to get the right biologic to the right cell in the right amount. Where does the RNA go once they go in the cells, right? And really, we've sort of only looked at vaccines for now, where there's only one example of mRNA based therapeutics right now that is clinically approved, but what else can RNA do, right? That are some of there's certainly a lot of opportunities for mRNA therapeutics, and I think it's going to be a really interesting field in the next few years. So with that, I would like to acknowledge a few folks. A lot of the work that I presented here was funded by Simons Foundation and NASA. A lot of the work was actually done in Phil Bevilacqua's lab at Penn State University, where I was a postdoc. So with that, I'd be happy to take any questions.
Thank you, Dr. Raghav R. For an excellent presentation about RNA and mRNA vaccines. It was very informational and explanatory presentation about RNA. And our next session, that is question and answer session, is open for all the participants. If you have any queries about the presentation or anything related to RNA, the platform is all of open for you. And also, you can also write the question in chat box. You can post.
So I see a chat question which has asked, I believe the question is, can we do RNA PCR for pathogen detection if possible? Why do RT-PCR, right, using thermostable RNA-dependent RNA polymerase? I think that this has actually been done, so you don't need the reverse transcription step. And I don't know on the top of my head what lab the work is from, but the short answer is yes, you can actually do RNA PCR without having to do RT-PCR.
Okay, I've got one question. For the ribo switching, does the external factor affect the function of ribo switching?
Yeah, so usually for the ribo switches, they tend to sense the metabolite concentrations, right? Like, for example, if in case of bacteria, if there is high concentration of, let's say, you know, a flavin mononucleotide or some metabolite that the ribo switch controls, then essentially at a high concentration, the ribo switch gets activated and turns off the gene that synthesizes that particular metabolite, so that the bacteria is basically using that switch to regulate the concentration of the metabolite.
Thank you.
Yeah.
Hello, Raghavji.
Yep. Sorry, तपाईँको sorry, तपाईँको इन्टरनेट एकदमै अनस्टेबल छ जस्तो छ।
जस्तै यो कोभिडमा ओके ओके
या तपाईँको इन्टरनेट अलि अनस्टेबल छ सो मेबी च्याटमा लेख्नु भयो भने आई क्यान सी इट।
There is one question from Ms. Nasla Sarke. She wants to know that how the byproduct of vitro synthesis of RNA by ribosome are removed. Can you answer this question, Mr. Dr. Raghav?
Sorry, the question was, how is the byproduct of?
She said how synthesis of RNA can be removed by ribosomes. I will write the question as posted.
So I think that there are obviously other
I've sent the question in the chat box.
Okay, yeah, sounds good. So there are obviously, so in the case of the membraneless compartments, because it doesn't have any membrane, the molecules can actually freely diffuse between the inside and the outside. So that's like, you know, you don't really, it's passive diffusion. So you can actually diffuse any molecule that you make or any molecule that is inside membraneless compartments. So whether it is synthesized by RNA enzymes or you know, any molecule that is within the compartments. I hope that answers the question.
I hope she got the answer. And there is another question from Mr. Poudyal. If you could.
Yeah, so I guess, you know, so yeah, Prajwal Raj Bhandari just asked if, well, it's more of a comment, I guess. It says many Nepalese scientists say classical way of making vaccine in Viro cells is far better than modern way of making vaccine like by Pfizer and Moderna. What is your thought on this? I think that there's really no right or wrong answer to this, right? I mean, clearly for the pandemic type situation, it seemed like mRNA based vaccine was, you know, it was quick because we could rapidly get the vaccines. You know, that certainly saved billions of lives. So I think it's clearly good for a pandemic type situation. Whether it would also be good for more endemic type situation, that is debatable, right? Like, because mRNA molecules are not very stable, so we can't really store them for a long time as things currently stand. So I think there's going to be pros and cons, and whether one's better than the other, I don't know. But certainly it seems like, at least in the discovery phase, where you want to test multiple multiple antigens, multiple biologics, whatever you want to express, it does seem like mRNA platform seems to be suited for this rapid discovery phase. At least that is at least my thought in this. But really, to give a short answer, I don't think there is one. I don't think one of the system has better advantage or the other. I think there is probably place for all kinds of different modalities.
We have a couple of more questions left. Time left for the couple of more questions. If anyone from participant, if you are willing to ask, you can.
You guys are very shy.
Don't be shy. Please feel free to ask the question.
Yeah, feel free to reach out to me through email, or you can find me in Twitter. I'm happy to answer any questions you may have later on as well.
I think we have no more questions.
All right.
Participants. Should I wrap up? From participant, want to ask question? So this is not for this, not for today. I excellent presentation and the discussion between the speakers and the participants today. So there is no more questions, so I can conclude the webinar.
Great.
It's been a pleasure being with you, all of you today. I'd like to. Yes, Miss. Yes, sir.
Oh no, yeah, go ahead. Yeah, thank you for having me. Really enjoyed talking about my science and interacting, and I hope feel free to reach out to me. I'm happy to connect.
I'd like to appreciate the effort and the time of our guest speaker, Dr. Raghav R. Poudyal. Well, hoping that you will have enjoyed and learned new things in our webinar today. And I will end the webinar series. Many if you could start in the present.